Welcome to The Explainer. Today, we're diving straight into the very blueprint of life itself. We are looking at exactly how raw DNA sequences get transformed into functional, real-world biological insights. Because, if you've ever wondered how scientists actually make sense of the billions of literal letters in a genetic code, well, you're in exactly the right place. We're going to break down this complex world of modern genomics and specifically focus on one incredibly powerful tool that kind of makes the whole shebang possible.
Okay, let's dive into this. Right now, advances in sequencing tech are moving at an absolutely breathtaking pace. We can generate these mind-boggling volumes of genetic data in just a matter of hours. But here's the huge problem we face. Raw DNA, just sitting there on its own, is pretty much useless to us if we can't accurately read and identify the specific regions that actually produce functional proteins. Think about it for a second. What good is having the most detailed map of life ever created if you literally cannot read the street names or find any of the landmarks?
So to tackle that, here is our roadmap for today. 1. The DNA data avalanche. 2. Enter the ORF Finder. 3. Decoding the reading frames. 4. Free science for everyone. 5. Real-world biotech impact. And finally, 6. The future of genomics.
Section 1. The DNA data avalanche, or drowning in genetic data.
Every living organism on Earth stores its genetic information inside DNA. That is a universal truth of biology. But here's the catch. Not all DNA is created equal. While we can now sequence entire genomes incredibly fast, and actually, fun fact, a single human genome takes up about 3 gigabytes of storage, organisms only use very specific targeted regions of this massive genetic data pile to actually encode the proteins that do the essential biological work. The rest of it? It's just not translating into the proteins that build cells or fight diseases or digest your food. So having the raw sequence just isn't enough. We have to find the specific parts doing the heavy lifting.
And this brilliantly illustrates the exact challenge scientists are facing today. Imagine reading a book that is millions of pages long, with 0 spaces, no paragraphs, and absolutely no punctuation. That is raw genetic data, massive and totally unpunctuated. What researchers desperately need is functional knowledge, those specific, identified regions that are responsible for producing functional proteins. We somehow have to get from that chaotic wall of data to precise, actionable knowledge, if we ever want to understand diseases or discover new genes.
Which brings us to Section 2, Enter the ORF Finder, the ultimate translation key.
First, let's quickly define some crucial jargon so we're all on the same page. What actually is an ORF? Well, it stands for Open Reading Frame. By definition, it's a potential protein coding region within a DNA sequence that contains a start codon and a stop codon. Going back to our giant book analogy for a second, if raw DNA is that endless string of letters without spaces, an open reading frame is like finding a capital letter that says, hey, start a sentence here. That's your start codon. And a period indicating the end of the sentence, your stop codon. Finding these ORFs is exactly how we turn a string of genetic gibberish into readable, meaningful biological sentences.
Now, finding these reading frames, it hasn't always been easy. In the past, the old way required really extensive laboratory analysis and just painstaking manual interpretation. Scientists would spend days, sometimes way longer, just trying to ID a single coding region. But look at the new way. ORF Finder has completely streamlined this entire process. It provides automated detection across massive DNA sequences, identifying potential genes in seconds, literally seconds versus days. It's an absolute game changer that bridges the massive gap between raw genetics and actual scientific insight.
So how exactly does it pull off this magic trick? Section 3. Decoding the reading frames and how the software actually works.
Let's move to this and see how it builds step by step. Step 1. The software examines the nucleotide sequences across all possible reading frames. It's essentially scanning the document from every possible angle. Step 2. It locates those crucial start and stop codons we just talked about, finding those capital letters and periods. And finally, Step 3. Once those regions are locked in, the software translates those sequences into amino acid chains. This lets researchers accurately predict the actual proteins that the DNA sequence might produce. The logic behind it is just incredibly elegant.
But predicting those amino acid chains isn't the finish line. It is just the starting point. The output from ORF Finder serves as the essential kickoff for a massive chain reaction of modern bioinformatics workflows. Once you have those predicted proteins, you move right into functional annotation to figure out what the gene actually does. You can do comparative genomics to see how it matches up with other species. You can even run evolutionary analysis. None of that advanced science happens without first running the raw data through ORF Finder.
Section 4. Free science for everyone. Democratizing bioinformatics.
So, the crucial point here is, you might think a tool this powerful would cost millions, or, I don't know, require a supercomputer to run. But no way. ORF Finder is a web-based app developed by the National Center for Biotechnology Information, the NCBI, and it is 100% freely available to the public. It requires no expensive computational infrastructure whatsoever. Because of this, it entirely breaks down financial barriers and empowers students, educators, and researchers all over the globe. Whether you're sitting at a top-tier research university or just logging in from a high school biology lab, you have access to the exact same world-class sequence analysis tool. Let's zoom back out to the macro level. Section 5. Real-world biotech impact. Transforming medicine and agriculture.
This is where the theoretical stuff becomes incredibly practical for us. In infectious diseases, scientists use ORF Finder to study things like antimicrobial resistance, which directly leads to faster, safer vaccine development. In oncology and genetics, identifying mutations within an open reading frame helps researchers see exactly how a mutation changes a cell's function. That paves the way for targeted therapies and precision medicine for cancer and inherited disorders. And in broader biotechnology, analyzing newly sequenced organisms lets companies identify genes perfect for recombinant protein production or even synthetic biology. Finding these tiny little reading frames quite literally changes the world we live in.
And I should really emphasize that ORF Finder doesn't just work in a vacuum. It is the ultimate team player. It seamlessly integrates into these much broader bioinformatics workflows. The protein sequences it predicts, they can be instantly plugged into sequence alignment tools, structural prediction software, and massive biological databases. It takes the raw DNA, translates it, and smoothly passes the baton to the next set of advanced tools so scientists can nail down protein function and evolutionary conservation. As we head into our final section, let's look at where computational biology is heading next. Section 6, the future of genomics, AI and beyond. Look, as biological databases continue to expand at just a staggering rate, the speed, the accuracy, and the confidence provided by ORF Finder remain the reliable, rock-solid foundation for all modern life sciences. It consistently empowers researchers to unlock the full potential of genomic data. Without this capability at the ground level, we would just be hoarding data without any real way to understand what it means. Now, what's really interesting about this is a point from researchers in the field. To quote, "With continuous advancements in sequencing technologies, artificial intelligence, and computational biology, the importance of efficient ORF identification will only continue to grow." Really think about that for a second. As AI gets smarter and sequencing gets faster, this core act of identifying reading frames doesn't become obsolete at all. It becomes even more vital. It's the necessary gateway for literally all future advancements in the field. Which leaves us with this final, thrilling thought to ponder today. As we use tools like ORF Finder to decode the foundations of life faster than we ever have before, what biological mysteries are we going to unlock next? Are we finally going to understand the genetic roots of complex diseases? Will we engineer entirely new biological solutions to massive global problems? The data is out there, and it is waiting to be read. Thank you so much for joining me for this explainer. I really hope you found this deep dive into the hidden machinery of genomics as absolutely fascinating as I do. Keep asking questions, keep learning, and I'll see you next time.